Morning, my dear students. Welcome to our Zoology class. In the last class, we studied about the fifth lesson, DNA replication. It's very important 5-mark question, and hope you all studied this question answers. And today we are going to study about one important topic. Today's our topic is transcription. DNA transcription. See, here the definition for this word transcription is the copying of genetic information present in one strand of the DNA molecule into a RNA molecule is called transcription. So that the process of copying the genetic informations from one strand of the DNA into a RNA molecule is called transcription. Understand of you? See, normally to produce a protein molecule, first the DNA is producing the RNA by a process is called a transcription. Then the RNA is involved in the another process called translation. Finally, the protein molecules are produced. So, the DNA is producing RNA by transcription. Then the RNAs are producing the protein molecules by translation. Understand a few? This is the normal process. But in some retrovirus species, retrovirus meaning example for retrovirus we can say HIV virus. So in retrovirus species, this transcription process takes place in a reverse direction. That means first the RNAs are produced. The RNA producing the DNA by reverse transcriptase. Understand? Then the DNA producing mRNA, finally the protein molecules are produced. So in retrovirus species, this transcription process takes place in the reverse direction or opposite direction. Understand a few? So I hope you understood the definition for this transcription. So transcription meaning the process of copying the genetic information from one strand of the DNA into a RNA molecule is called transcription. Understand a few? Then, see, there are different types of RNAs are present in the cell. mRNA, in we can say messenger RNA. mRNA meaning messenger RNA. See, here mRNA meaning the RNA molecule which carrying the genetic information to encoding a protein molecules from a gene into a cell is called mRNA. So the RNA molecule which carrying the genetic information encoding the protein molecules from a gene into a cell is called mRNA. Understand a few? Then for this transcription process, for the transcription process, first the double helix DNA molecule should be pulled temporarily for a short period. So listen very carefully. For this transcription process, a double helix DNA molecule should be pulled temporarily for a short period. After that only, the RNA molecules are produced by a enzyme is called RNA polymerase. So I hope you understood this point. So RNA polymerase enzyme is one of the important enzymes. This enzyme is only responsible for the production of RNA. Understand? So for this transcription process, first the double helix DNA molecule should be pulled temporarily. Understand a few? Now, the nucleotides present in the newly synthesized RNA molecule is complementary to the nucleotides present in the DNA molecule. So the nucleotides present in the DNA is complementary to the nucleotides present in the RNA molecules. Understand a few? Then the next point, say two strands are present in a DNA molecule. You all know that. Normally DNA is a double-stranded molecule. Say both strands of a DNA molecules are not copied at a time during the transcription process. So during the transcription process, the genetic information present in the both strands are not copied simultaneously because of two reasons. So because of two reasons, the genetic information are not copied. The genetic information present in the both strands are not copied simultaneously. Understand a few? The first reason is, say, if both strands of a DNA molecule act as a template, they will produce different types of protein molecules, and these protein molecules has different types of amino acids. Understand? So, if the both strands of the DNA molecule act as a template, they will produce different types of protein molecule with different types of amino acid sequence. Understand? So finally, from each and every fragment of a DNA molecule can produce 2 or more protein molecules. It is very complex. Okay? Understand? So this is the first reason. And the second reason is, if two mRNA molecules produce at a time simultaneously, they will complementary with each other. Understand? It leads to the prevention of this transcription process. So because of these two reasons, the genetic information present in the both strands are not copying simultaneously. Hope you understood this point. So once again I repeat this last point. Because of two reasons, the genetic information present in both strands of a DNA molecules not copying during the transcription process. The first reason is, if the both strands act as a template, they will produce different types of protein with the different amino acids. And the second reason, if two RNA molecules produce simultaneously, they will complementary with each other. So it prevents the transcription process. Hope you understood that. Next, transcription unit. Listen very carefully. Transcription unit. See, a transcription unit means the place where the transcription process takes place. Okay, so the transcription unit has three important regions. One is a promoter region, and the second one is a terminator region, and the third one is structural gene. So the transcription unit meaning the place or the site where the transcription process takes place is called transcription unit. We have three regions: promoter region, terminator region, and structural gene. Understand? Then see, the promoter region is located in the 5-end. You all know the structure of DNA. It has two regions, 2-end. Just imagine this is the 5-end, this is the 3-end. So this promoter region is present in the 5-end of a DNA molecule. Understand? Then this promoter region is only responsible for the binding site. That means this promoter region only acting as a binding site for the DNA polymerase. Listen very carefully. So this promoter is one of the region or one of the site present in the 5-end of a DNA molecules. So in this region only the polymerase enzyme, okay, the polymerase RNA polymerase enzyme is binding in this region only. Understand? So this end, this 5-end or this promoter region is acting as a binding site for the RNA polymerase. Understand? Then next one is terminator. This terminator region is present in the 3rd-end. Terminator region is present in the 3rd-end. Understand? Then this terminate region only stopping the transcription process. Stop the transcription process. Hope you understood this point. So yes, the transcription unit has three important regions. One is promoter, second one is terminator region, and the third one is structural region. Promoter region is present in the 5-end of the DNA molecule, and the terminator region is present in the 3-end. This promoter region is act as the binding site for the RNA polymerase enzyme. Then this end, 3rd-end, is acting as a terminate or the stopping region of this transcription process. Then the 3rd region, so promoter region over, terminator region over, and last one is structural gene. So listen very carefully. The structural gene is two types. It is divided into two types. Okay, in eukaryotic organism, the structural gene present in the eukaryotic organism is called monocystonic structural gene. In eukaryotic cell, the structural gene present in eukaryotic gene is called monocystonic. Yes, SG means structural gene. So the structural gene present in the eukaryotic cell is called monosystemic structural gene. Then the structural is present in prokaryote. Prokaryotic cell is called polysystemic. Polysystemic structural gene. So promoter region already told you, this region is present in the fifth end of a DNA molecule, and the terminator region is located on the third end of the DNA molecule. The structural gene, there are two types of structural genes are present in the transcription unit. In eukaryotic organism, name of the structural gene is called monosystemic structural gene, and in prokaryotic organism, this region is called, the structural gene is called polysystemic structural gene. Understand a few? Then here, this monosystemic structural gene, okay, carrying only a single gene. Okay, so this monosystemic structural gene carrying only a single gene responsible for a single protein molecule. Single gene carrying for a single protein molecule. But here in prokaryotic organism, already I told you, polysystemic structural gene is present. So this polysystemic structural gene carrying many genes, a cluster of genes called operods. Okay, so a cluster of genes are present in the polysystemic structural gene, and this cluster means a group. Okay, group of genes are carrying a single protein. Hope you understood. So in eukaryotic organism, monosystemic structural gene is present. This gene carrying a single gene only, and this single gene only responsible for a single protein. Understand a few? Then in polysystemic structural gene, there are a cluster, that means a group of genes are carried present, and all these genes are togetherly producing a single protein molecule. Understand a few? Understand a few? Then, see, in eukaryotic cells, the promoter region has more amount of adenine and thymine region. Listen very carefully. In eukaryotic gene, okay, in eukaryotes, the promoter region, some region of the promoter has more amount of adenine and thymine, AT region. Okay, highly AT regions are present. Okay, and this region is called Tata box. GA box. Understand a few? Then, so this Tata box is present in eukaryotes. The same region present in the prokaryotes is called Pricknow box. Once I repeat this point, see, in eukaryotic organism, eukaryotes, some region of the promoter has more amount of adenine and thymine, AT region. High AT regions are present in some region of the promoter region of the prokaryotes or eukaryotes. So this region is called Tata box. And the same region present in the prokaryotic organism is called Pricknow box. That's not few. So it is one of important 2-mark question. That's not few. Then, so now we completed the transcription unit. 3 regions present in a transcription unit. Then, listen, say, always the DNA dependent RNA polymerase enzyme catalyze this reaction, transcription reaction, from 3 to 5 end. Okay, from 3 to 5 end. So this end is called template strand. This end is called template strand. So 3, 5. Okay, so the DNA dependent RNA polymerase enzyme catalyze or speed up this transcription reaction from 3 to 5 end. So this strand is called template strand. Understand a few? Then the opposite strand, that means 5 to 3 strand. Okay, so this opposite strand is otherwise called coding strand. Okay, so this opposite strand is called coding strand, and in this type of coding strand, this strand is displaying during the transcription process. Understand a few? So listen very carefully. So this strand, so the 3 direction strand is displaced during the transcription process. So this strand is called coding strand. This strand is called a coding strand. Okay, and the DNA dependent RNA polymerase catalyze this transcription reaction from 3 to 5 strand, 5 end strand. So this strand is called terminal strand. Understand a few? So I hope you understood this part. Then next part, the RNA polymerase. Already I told you, is one of the very important enzyme playing a very important role in the transcription process. So this enzyme, this RNA polymerase has two important regions. The first region is called coenzyme. Okay, the first region is called coenzyme and the second region is called sigma subunit. Okay, listen very carefully. The RNA polymerase enzyme has two regions. The first region is called coenzyme and the second region is called sigma subunit. Now listen very carefully. This RNA polymerase, the core enzymes of the RNA polymerase enzyme only are responsible for RNA synthesis. Okay, so the RNA polymerase enzyme has two regions, core enzymes and sigma subunit. These four enzymes only are responsible for the synthesis or the production of RNA. Some of you. Then this region move ahead, okay, and leaving this sigma subunit behind the promoter region. Behind the promoter region. Listen very carefully. This four enzyme move ahead and leave this sigma subunit region behind the promoter region. Understand a few? And this region, sigma subunit. Okay, the sigma subunit is responsible for the recognition of promoter region. Recognition of promoter region. So listen very carefully. The RNA polymerase enzyme has two important regions. One is core enzyme and the second one is sigma subunit. This core enzyme region is very important. That means this region only responsible for the synthesis of RNA, and this region move ahead and leaving the sigma subunit behind the promoter region, and the sigma subunit is this region is responsible for the recognition of promoter. Stand up here. Then the last point about this transcription unit is the transcription sequence. Okay, form a hair-like structure. Okay, form a the terminator region. Okay, not transcription. The terminator sequence of a DNA molecule form a hairpin-like structure. Okay, and this terminator sequence also require a additional protein. Name of that protein is called RHO protein. Okay, RHO protein. So RHO protein is the protein molecule, additional protein molecule present in the terminator sequence. Okay, and this terminator sequence finally forming a hairpin-like structure. Understand a few? Understand? So I hope you all understood this transcription unit, structure of transcription unit. Understand a few? Okay, my dear students, I hope you all understood these question answers. Okay, study today itself. Many 1 mark questions are there, and it's a very small 5 mark question, but many 1 mark questions are there. Okay, so study well and send to the answer to me. We will see in the next class. Thank you.